ahead and start. And as I always do with courses, whether I'm teaching undergraduate evolutionary biology for hundreds of hours, it seems like, or whatever else, I try to remind people where we've been each time. So you'll see me doing this every time we meet, all 8 sessions, so I apologize for the repetition. Oh, that's the other thing. I was mentored to teach by telling people something, telling them what I told them, and reminding them what I told them. So you'll see a lot of things repetitively here, and that's on purpose. I'm forgetful, but I'm not that forgetful. Okay, so that's why you'll see a lot of material that's actually repeated, just like evolutionary processes. So I'll remind you that yesterday in our 2 sessions then we talked about how do we define species, a few suggestions, a few approaches that people have proposed, some classical like Ernst Mayer's, and Joel Craycraft's, I would say, is classical as well, or applied quite frequently, I should say. Maybe that's the better way to say it. And so the PSC, phylogenetic species concept, and the BSC, one that people don't know about as much, but I like because it, like we talked about, remember, it really tries to draw as much information as we possibly can to give us an idea of whether or not we are dealing with different lineages. For whatever reason we need to know, whether or not it's systematic taxonomic reasons, or whether or not it's trying to understand, like in my case and other people's case, the processes that lead to evolutionary diversification. And that's Alan Templeton's cohesion species concept. And then we talked about the fact that Charles Darwin actually didn't have one. He said that they were artificial. Species were artificial like genera. And so that was his take. And we talked about why he might have concluded that, both with biological, quote-unquote, scientific reasons and rationales and evidence, as well as the philosophical stance that he took towards special creation, okay? And so then we moved, obviously, into session 2, or next into session 2, and we talked about modes of speciation. In other words, these units, however we define them, how do they evolve? How do they come about? And we started out, once again, with a paper by Ernst Mayr, which we will again in a few moments. And we talked about the fact that because of reproductive isolation, because of a number of assumptions that Mayr had, along with that biological species are defined by reproductive isolation, period, that's it, that it required allopatric speciation. It required allopatry, for speciation to occur. But then we talked also about sympatric speciation. We didn't really go into Sarah's paper or Guy's paper too much, but we talked about the fact that sympatric, in fact, when we were up here, if you recall, we talked about sympatric speciation, how speciation might actually occur through disruptive selection, building up of linkage disequilibrium, etc. And so we'd already actually discussed it up here, is why I didn't emphasize it down here. And then we went through reinforcement speciation. We talked about the problems with that model and how it might work in nature. And then we didn't actually talk about Doug and Pam Soltices, or the Solti, as a bunch of us call them, and their work, but we will be talking about allopolyploid speciation throughout the time that we're talking actually about through my book and you guys reading in those chapters. And then finally, we also talked about Dolph Schluter's ecological speciation model and what it might require. And of course, we talked about his sticklebacks and some other observations, more recent observations. All right, so concepts of species, how they might arise. And today I want to, this morning, with the time that we have, I want for us to work through what the pattern of speciation might be. Darwin envisioned it as long, okay? That it took relatively a long time for one form to descend with modification into another form with branch-like tree-like aspects to that, okay? And that you would have incremental, small changes along that line. In fact, they would be imperceptible in the fossil record, okay, if you looked at it, at least until you had reached some kind of a change where you could detect it in the morphology, but many of the changes would be so small you wouldn't notice them, okay? What we're going to talk about today is whether or not that is actually the pattern that is seen in the fossil record. We're going to talk about why the pattern that is seen in the fossil record may be seen, all the way from biological evolutionary reasons to is it error, okay? Is it missing data? Is it whatever else? Okay, we'll talk about that, and we'll use Gould and Eldridge to do that. But we'll also talk about a model by Ernst Mayr where he talks about genetic revolutions, and we'll start out with that, and about how these may relate, Ernst Mayr and Steve Gould's and Niles Eldridge's model. And then we'll move on into more recent papers, but not yesterday, in terms of Axel Meyer et al. Out of Alan Wilson's lab, and then one by Scott Hodges and I on a plant group that Scott's been working on since he was in my lab as a postdoc in the early 90s. And this is really Scott's work, okay? He works on Aquilegia. He had a Nature paper not that long ago summarizing a number of different things. He does beautiful work, absolutely gorgeous work. And I was fortunate, was very, very blessed actually to be able to work with him and to do some work with him, but I'll tell you some anecdotes about that. And then finally, depending on how we're going, I'll either present a part or all of a PowerPoint presentation on this topic of rapid evolution due to natural hybridization or in general genetic exchange, or we'll go through this paper and talk about it through this book chapter. Okay, so Ernst Mayr proposed what? You guys hopefully have read this, and let's see, I never remember what the numbers are. Let's see, that's it, 7946, just give me a second. Isn't it great when your instructor is so well organized? So, Ernst Mayr's book chapter, and what did he mean, I guess? Let's just start it out with me asking you a question. Maybe you could just summarize what he's talking about here in a few points. But what does he mean by change of genetic environment and evolution, do you think, from what you read? Remember, we're taught, this is in the general topic area of tempo of speciation. How fast, how rapid, you know, can speciation be, can evolutionary diversification be? Remember Darwin's idea of long periods of time, and actually Ernst Mayr's idea of that as well. In other places, not necessarily in this book chapter. Okay, questions, comments, but what does he mean here? What has he told us in this book chapter? Please don't be shy, and get over your sleepiness quickly. What is Ernst Mayr's points, some of his points? Okay, it's allopatric. Thank you for picking up on that. Because one of the things that we could ask in this particular model is whether or not it's allopatric or parapatric or whatever else, okay? It's allopatric, according to Mayr. But we will talk about how it's also, this will make him roll around in his grave, how it's also sympatric / parapatric. Yeah?
Just thinking, does it talk about the extent of the hybridization zones and how slowly, like, for how long it stays?
Mm-hmm.
Hybridization zone.
Yeah, and I agree with you. However, keep in mind that he would never have he was not discussing this model or proposing this model in the sense of hybridization. Okay? We're going to walk through the steps that he discusses in a moment. But I would argue, and others, I think many others now would argue, that he did propose hybridization as a portion of this model. But he did not propose that. Ernst Mayr felt, let me just explain to you how he defines this and how he defines the importance of hybridization, and we'll see this in quotes that I'll give you in some of the PowerPoint presentations. The way he defined hybridization, and other people did too, was hybridization was when you had crosses between different species. Okay? By definition, biological species cannot mate, cannot cross. Therefore, hybridization cannot occur. If you have crosses between well-diverged subspecies or any other subspecific category, really different organisms, that is not hybridization. That is within a species, and so that's just gene flow. So do you see how circular reasoning in a lot of ways defined hybridization, any kind of evolutionary importance of hybridization out of existence? It became best an epiphenomenon, but usually it was non-existent because, well, we know species can't hybridize because species are reproductively isolated. So it comes back on itself. So that's why I say that what we're going to talk about in this model, I'm going to argue is exactly, explicitly hybridization. But he would not, absolutely would not have argued that. I don't want to misrepresent what he was arguing. Okay, so what else? Anything else? I just wanted to draw up on the whiteboard what his steps were in this model. Anything else that you gleaned from this chapter? By the way, I think it's a fundamentally important model. I think actually it's very reflective of how evolution occurs now that we're getting genomics in particular, how we see gene divergence with genetic exchange. I think it's absolutely an excellent model, regardless of how he perceived it. Yeah.
Is it somehow also linked to ring species?
You know, he didn't really link it to ring species per se. You want to define what ring species are, though, for someone else who may not have heard of them? What's a ring species? Complex, generally, is what we're talking about.
So when you have like a species, like a range of species distributed over a large geographical area,
Mm-hmm.
so the farthest the two species are like diametrically, they are more distinct from each other,
Okay.
compared to the ones that are more forward.
Yeah, that are closer geographically. Yeah, so Mayer's model has been tested, and I want to actually talk about this a little bit later anyway, so imagine that this keeps going around, and actually we can just keep doing it. And so if the evolution of this ring of divergent forms began here, okay, and actually had a directionality to it, by the time this gets back here, these cannot cross. They're reproductively isolated. And actually, some of these may be reproductively isolated, but his proposal would be that they actually increase in reproductive isolation as you go around because of time and divergence over time. There are actually some groups, he proposed it obviously for seabirds and things like that, I believe. So he was an ornithologist. But anyway, it's been tested in some groups and falsified, but it's also been supported recently in a group of either, shoot, I should remember this. Yeah, but there's another one, isn't there? I thought there were two. One of the people in Holland, the Netherlands, also was working on something. Anyway, but yes, it has been supported, inferred to be accurate for some and falsified for other groups that he suggested were ring species. So in answer to your question, not really in this model, but it's close in some ways. But anyway, it has components that are similar to this. Well, let's go ahead and write out the steps, if you will, to this. So let's draw. So you have a large range of a species. Okay? Start with this. So that would be step 1, if you will. Step 2 is that you have peripheral populations. And this is critical, and this is why when you said it's allopatric speciation, or allopatric, I said thank you. Because a lot of times we miss this, but we have to put it in the context of what Ernst Mayr would have been thinking, which is sympatric speciation doesn't work, right? I mean, functionally it's impossible because of gene flow if you have sympatric. So anyway, you have a set of peripheral populations out here. Okay? Now, what happens in these peripheral, small in size in terms of N sub E, geographic range 2, compared to this. But what is happening out here that's not happening in here, according to Ernst Mayr? Genetic drift, okay? Sampling error. So his argument is out here you end up with genetic drift. And we'll talk about where he got his ideas for this in a few minutes. So you have genetic drift. Now, genetic drift does not always lead to new combinations of alleles put together, right? I mean, he's saying you have mutation out here in these different populations, and through drift, right, through drift, inbreeding, and extinction. So many of these are expected, on average, to go extinct. But occasionally, according to his model, occasionally you will end up with novel genotypic constitutions in some of these peripheral populations. Now what does he propose happens? What's the next step, the next part of his model? Like I say, this is a fundamentally important model for punctuated equilibrium. When we'll talk about it, Steve Gould and Niles Eldridge have used it to explain the process that may underlie punctuated equilibrium in the fossil record. But it's also really important for us to understand how, in some ways, how cichlids might be given rise to and other things in adaptive radiations. Okay, so what happens here? Did you pick up on that? What happens between A and B? Okay, they're developing different genetic environments, but what happens once that's developed? What's the next step in the model between these two populations? What do? They cross, okay? They have a gene flow event, whatever you want to call it in Myrian terms in his model. But these mate. They encounter one another, overlap, and form hybrids. But he didn't call them hybrids, okay? Not really. And so that leads to novel, in some cases, genotypic arrays leading to what he said were genetic revolutions. Okay? Right, now what might happen according to his model? We have this now. What would he propose, or what did he propose now? What would be the next step potentially here? There is increased selection, that is correct. And once again, these novel genotypic arrays, this genetic revolution, could lead, this increased selection could lead to extinction of this. But if it doesn't, what do we potentially end up with? Remember, this is a model of how biodiversification occurs.
Does it occupy separate tissues?
Yeah, it can. They can then spread into new niches. Okay. And that could lead to new species. What else could happen, according to Meyer, is that this could actually have an increased fitness in here. And it could drive this to extinction and replace it. Okay? There's different outcomes that he proposes. This is a word model, so there are different outcomes of this that he proposes. Okay? So what I want to remind you of is what I said a little bit ago in this model. Ernst Mayr never called this hybridization. He did not say that this was natural hybridization was affecting this. This was merely gene flow events within a species between groups of different genotypes. Okay? With our new methodologies that we have for measuring genotypic and genomic variability, and with the new understanding of what genetic admixture looks like, this, in today's modern terms and in our modern understanding, would be called hybridization. And also it could be called hybridization because now we're allowed to call it hybridization from a pragmatic standpoint. Before, during 1954, I think is when this came out, people were not allowed to call it hybridization because everybody knew that sympatric speciation and hybridization were not important, especially, of course, in Ernst Mayr's animals. Remember that quote that he gave when he was 100. He still held that allopatric speciation was the way that everything evolved in terms of animals, quote unquote higher animals. Okay? So, but this kind of a model is exactly what is very similar to what Ola Seehausen and others have proposed for hybrid swarm adaptive radiations in all kinds of plants and animals. This is the kind of model that many of us have proposed and tested for in homoploid hybrid speciation. This is the kind of model that actually fits allopolyploid speciation in many cases, particularly where we see, like the Soltices talk about, subspecific crosses leading to new species, right? So this model, like I say, is actually very applicable to many evolutionary speciation kinds of events. Very, very applicable to what we know now. Ernst Mayr, I don't think, saw that per se, but he was very perceptive in what he was proposing. Now, does anybody know who, I've mentioned this already before, do you have an idea of whose work he might have been basing this genetic revolution, genetic drift components on? Say one more time. Yeah, it was, well, Sewell Wright. Yeah, okay. So Ernst Mayr and Dobzhansky and a number of them were really taken, for a while anyway, with Sewell Wright's work. Okay? And Sewell Wright suggested that in fragmented populations, you had genetic drift, right? repeated bouts of genetic drift in small populations, and then gene flow events between populations at different or occupying different adaptive peaks. Okay, and those gene flow events then could lead to novel genotypes and occupying, again, novel adaptive peaks. Novel adaptive peaks. And so that's really, honestly, what Ernst Mayr is picturing here. Except he's doing it in a geographical sense, and none of Sewell Wright's models are explicitly geographic. They're on a multi-dimensional calculus landscape. Okay? And if you've ever seen that drawing that, like concentric circles of like isobars or isoclines, and then of Sewell Wright's model that he drew out, well, and then if you've read anything about his work, you really can't get there from his math. You have to, first of all, think thoughts that I can't think in terms of the kinds of math that he did. But basically, this, though, was overlaying onto this in this model. And Ernst Mayr recognized that. He jettisoned Sewell Wright's ideas later because people accused Wright of just really emphasizing, how I emphasize natural selection and adaptation. They argued that Sewell Wright was way overboard, arguing that genetic drift could do everything, which he never really did. He always had these other components. He just emphasized in sub E contractions of populations or smaller populations and the kind of changes that can be in those small populations. And just as an aside, it made sense. You remember Darwin's non-species concept made sense for what he worked on and what he was thinking about. Sewell Wright's ideas here were because he was a breeder. He worked in animal breeding. Okay, and in fact, his formula are still used by animal breeders and plant breeders worldwide. We may not know we're doing that, but we are. And so he came up, of course, with FSTs and all these kinds of, he was the author of those. And so his idea in animal breeding was line breeding, inbreed, 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 take two lines and cross them and look at all the really marvelous things you get out. Some are trashy, you can't use them. Some are really fantastic. Okay, just like Darwin used pigeon breeding or whatever else kind of breeding to demonstrate natural selection by looking at artificial selection. Anyway, that's an aside, but what I'm saying is this kind of conceptual framework, conceptual? I just switched to my southern accent, conceptual. So anyway, that kind of a framework is what then Mayr borrowed to talk about how speciation could occur in a geographic sense. Does that make sense? It's a cool model, actually. I think he was really wrong about the kind of allopatric versus non-sympatric at all, but really set up a lot of us for thinking about how our models of evolution might work. Yeah?
What you told made sense, but Mayr did believe there is nothing else allopatry. Now here we see there is a main population, all the subpopulation small, so it is more times peripatry. So again, like there was no habitation in his species. So here habitation also is happening. So how all these things, even peripatry, even habitation, he explained with allopatry.
Well, the way that he explained it, you're talking about if he's got this going on, how does he call it allopatric? Well, because when all this evolution is going on, these are separated from this. They're separated from one another as they're going through genetic drift. And these are not different species. Hybridization was defined as crosses individuals between different species, within different species. By definition, this wasn't that to him. Mainly because circularly he would say, well, species can't cross. It's like, well, but anyway, so that's part of it, but that's why. He said his Perry-Patrick model was an allopatric model. I just don't want to misrepresent. He proposed it as an allopatric model. I would propose this as a classic adaptation of a sympatric model. You know, where you have hybridization and new hybrid species forming, that sort of thing. That's classic. But we know that now. We need to give him his dues, which is in 1954, we didn't even know what the genetic material was yet, right? So we need to put all of this in the context of he's still a giant we're standing on. I don't want to give you, we wouldn't be reading 3 of his papers if I thought that it was worthless, right? I would just tell you, no, we're not going to cover that because I just don't think it's worthwhile. Ernstmeyer gave us, look at the ring species concept. I think he was right, in a number of different groups on this. And he did all of this from looking at skins, right? Feathers and morphological characteristics. That takes enormous biological intuition. Really seriously, I worked 6 years as a naturalist, as a mammalian phylogeneticist, systematist, and I am not good at it. I just don't have a lot of biological intuition when I'm looking at these things to go, wow, I just don't see the differences. With genomics, I'm a little bit better, but with the morphology, that's what he had. And yet he figured so many things out, and I think he was exactly right about this. From what we see now with many, many different data sets, I just think his conceptual framework was incorrect. I think he was so locked into species as almost deified units, honestly. And people accused him occasionally of being a creationist, just to, I guess, rile him up. He was not. And but his species, could bump up against one another like rocks, but they couldn't really exchange any material. It was really a philosophical stance, okay? It really was, rather than a scientific one. But that aside, this model still works, whether he calls it allopatric or not. Yeah. Yeah, so in his model, these are going to be separated from each other and because remember, if they're not separated, it's not allopatric. Well, peripatric, yeah, exactly. I would call peripatric speciation, by the way, as but I think of peripatry because of how I was trained, especially in Australia, as a special case of sympatry, because they're still exchanging genes. But whatever we want to call it, but yeah, these are separated. In this model, these are separated. And the only time they actually come into contact is here, in his model, right? So, say again. Yeah. That's why I think this is generally applicable. That if there is divergence with genetic exchange is the rule. We know it is in viruses, prokaryotes, plants, is the rule, with allopatrics or allopolyploidy and all. But if it's a rule, then this model is exactly what we talk about with divergence with genetic exchange, right? Because these are separated. Imagine glacial refugia in North America. That's how I think of this. And then they come back together. Their zones. Now, people will ask, why aren't I a real champion, once again, of just purely sympatric speciation from within here? And it's not we talked about it yesterday. It's not that I'm against it. It's just that I feel like this model fits what we see in the genomic data. In other words, migration events scattered throughout a network over time as you go from earlier to more recent. And so we don't see continuous gene flow estimates. They may have been, but we don't. We see episodes of gene flow, which would suggest to me allopatry, parapatry, or sympatry. Allopatry, parapatry, sympatry. And also I'm thinking biogeographically, paleobiology, paleogeographic kinds of distributions that we know organisms that we've worked on had. Birch trees that one of my PhD students worked on, that is, both species must have co-occurred in far southern, southwestern US, and now only one of them is down there, and the other one is all the way, the closest populations of it are up in northern US. So, thousands of miles away, basically. And that's happened in the last 12,000 years. And the reason we know they were down south together is because we still have hybrid populations down there. And so that's what I'm thinking of, is why I think this model is applicable to quite a few things, if not almost everything. Great points. Other questions, comments? Mm-hmm. Mm-hmm.
So that's because he was mostly concentrated on allopathic mode.
Right.
So in whatever, by whatever driving force this happens, this battery without actually geographical isolation, this
Right.
process still can go on.
Yeah. So let's put this extension on it. You understand this is why, like yesterday, we talked about disruptive selection. Could the adaptive radiations have a component, a lot of disruptive selection associated with it? And I said the models do not, the original models are not talking about that. But I think that that's exactly what's likely to be going on, because we now understand that there's probably a lot of genetic richness there that's going to get sorted out through disruptive selection and balancing selection and all. But the original models didn't argue that. What I want you to keep in mind is his original article, his original model, his idea is no sympatric speciation. Before I do what I'm about to do, and suggest that maybe an extension of this would be quite interesting to think about. And that is, what if instead of having true allopatry, we have parapatry? So we actually have connections here between some of these, okay? And we have environmental differences here, right? A stepcline reflecting, like said, and Huxley said, this reflects a tracking of environmental change. So a tracking across here. I mean, why couldn't we have limited gene flow between here and here, and a buildup of genotypic variation here that is not here? So here and here, because of environmental selection, we're tracking the environment. Let's say it's elevational, who knows? But anyway, you have a parapatric situation. These then, okay, these, even within, say, this population to here, we could be walking across this way and see that, right? Because of a gradient, an environmental gradient. And then the crosses could be between those. Really, all we're requiring here for this model to work, for his genetic revolution, is to have some kind of a bottleneck, if you will, but also just a building up of genotypic differences and then crosses to give you novel. Basically what you're doing again is you're now, over time, you're building up different pools of standing genetic variation. different combinations of alleles, and then you're mixing them together again. I mean, that's what he's proposing with his allopatric. It's not different from that, what I just said. His is different, though, because it's allopatric. You would not be able to have this connection according to him, because then you would homogenize those two because of the trail of gene flow that's going on. So anyway, could we, does this make sense? Absolutely. And in fact, in many cases, this is what we infer from, once again, biogeographic information, genomic information, say chimpanzees and gorillas in Africa. The different species of gorilla have diverged with genetic exchange, the two species. They're no longer that, now they're allopatric because of habitat loss due to us, okay, due to our species activities. But the idea there was that there was a long period of continual gene flow, estimated, okay, hundreds of thousands of years probably, of gene flow. So it's really this, with them diverging in a more parapatric situation. Same way with the different kinds of different forms of common chimpanzee. So there are examples of this kind of a model, potentially, but parapatrically. Yeah?
If we actually have different species, like hybridization is leading to different, I don't understand why that will also give rise to punctuated because of speciation.
Why would it lead to punctuated equilibrium? Okay, so can we move on to Gould and Eldridge, and we'll flip back and forth. This is why I put these two papers together, is because Steve Gould, Niles Eldridge applied this model to try to explain their observations from the fossil record. So let's look. We're not leaving this per se, but let me get back here. Let's see, where are you? Did it? That's it. Okay. So, what is punctuated equilibrium? Somebody define it for us. Where is it observed? Okay. And that's the punctuation. What is the equilibrium? Yeah. So what happens during the equilibrium part? So you have punctuated periods, rapid evolution. What's going on in the periods between those? It's up there on the board. Yeah. Yeah. Stasis is what they... So the equilibrium part, these are data. They're not hypotheses. So where did they get their data from? This is Steve Gould, Niles Eldridge. Where are they seeing punctuated equilibrium? What kind? Yeah, but where are these organisms? Where are these samples from? Fossil record. Okay, I know you knew that, but let's just remember, we're not talking about extant taxa. Okay, we're talking about fossil sequences as you run from more ancient to more recent. Okay, so you said rapid, relatively rapid, relatively short time periods, evolutionary change. That's a punctuation event then, right? This is the equilibrium. Equilibrium, punctuation. Equilibrium, punctuation. Equilibrium, punctuation. Okay? So, let me tell you a story to emphasize this, and this generally gets questions. So, I came to the University of Georgia as a brand new assistant professor in 1989. I've already mentioned this. And I had to teach an undergraduate course, and they had me teach undergraduate evolutionary biology, which I've taught most years from then on, okay, including this coming up spring. And so I was in charge of the macroevolution part. We divided it that way back in the day. One of Theodosius Dobzhansky's former graduate students, who's a member of the National Academy of Sciences now, Wyatt Anderson, co-taught with me. He taught the microevolutionary part. Terrified me. I had Theodosius Dobzhansky's student, and I was teaching evolutionary biology with him. Felt really stupid. So anyway, so I'm teaching the macroevolution part, and part of it is the fossil record, okay, the biological record. So we talked about mass extinctions, et cetera, et cetera. And we were using a textbook by a colleague of mine, Doug Fatuma, who's at Stony Brook. And Doug is a famous, absolutely brilliant evolutionary biologist, works more on evolutionary ecology, ecological genetics. Doug wrote in the textbook that we were using that, this was 1989 now, remember, that evolutionary biologists were still debating whether or not punctuated equilibrium or gradualism was the most common observation in the fossil record. Okay, gradualism like Darwin, going from one to another type, sort of linearly, with small incremental changes over time, versus that. So anyway, that's what I taught. I said, look, it's still debated. Doug Fatuma, your textbook writer, says that. I don't know anything about paleobiological information, and so I'm just going to assume that that's correct. And by the way, I really respect Doug. Brilliant, brilliant guy. I went to a meeting in 1990 in Europe, and there was a keynote being given. I was given a symposium talk there, and there was a keynote, though, given by a paleobiologist, very famous one. And Jeremy was from Smithsonian Tropical Research Institute at that particular point in time, but he was moving to the University of Chicago. Very, very, very well-known international kind of guy in his work. And so he got up and he was talking about the fossil record, and he looked out in this audience, which was mostly people like me, i.e. microevolutionary kind of people, process-oriented folks. And he said, I need to say something to all of you here. And he said, I don't understand why people who do not work in the fossil record suggest ever that gradualism explains most of the patterns that we see in the fossil record. He said, it is very rare that we see continuous slow change over time. He said, that's what we see, period. He said, story's over. Punctuated equilibrium is not a hypothesis, it's an observation in the fossil record. Okay, that's 1990. When Gould and Eldridge published this paper, which, by the way, I think is one of the worst written papers in scientific history, which just blows me away because Steve Gould was such a brilliant, popular writer, okay? Just a really, really amazing communicator. However, this is a classic paper, so you had to read it. Sorry about that. I had to read it too. So when they came out with this in 1977, though, people started talking about something to make fun of them. And so we'll talk about what they meant by this. Oh. Right. Okay. That. So I have a little, I want to leave this up here while we talk. What people argued that they were wrong. Okay? There was a 1980-ish Evolution Society for the Study of Evolution meeting in the U.S., and it was apparently a bloodbath, with people calling them all sorts of names when they presented this paper, and and all this sort of stuff. It really got ugly. It's also funny because scientists, of course, fight like that, so it must have been hilarious. It's not a bloodbath like they could actually hurt one another. And so what people argued was that what they were suggesting with this kind of rapid evolutionary change, those punctuations, was something that was called a hopeful monster. I think I'm misspelling this each time. Probably misspelled Sewell right. I can't remember if it's a double E or an A. I apologize. So, hopeful monster. Okay? People argued against it and said, your model can't work because this proposes a hopeful monster. This rapid change from here, from this type to that type. Does anybody know what they meant? It was a terminology that was applied and talked about by Goldschmidt. It's actually important because then we'll go into this to see how they suggested they could counter that. Does anybody know anything about this? It's not a terminology we use much anymore. A hopeful monster, what they meant by that was, what Goldschmidt meant, was that evolution did not occur mainly by the buildup of small mutations of small effect. We would call it that now. But rather that you had macro mutations that gave you jumps. And he pointed to things, he worked on insects, he pointed to things like butterfly wings, spots, and all these sorts of variables that you could see these jumps, quote unquote, from one kind of morphology to the next with no intermediates in real time. And also large mutations that you could see in the lab. Drosophila with too many wings, okay? Or too many eyes, or too few eyes, or whatever. So he was pointing to, he said, that's why there are gaps in the fossil record, for example, that Darwin observed. He said it was because there really were jumps. There weren't any intermediates. So people argued that that's what Gould and Eldridge and Gould were arguing for. And this was known, quote unquote, known not to be possible. Macromutations were not possible. Micromutations in populations over long periods of time were what drove evolutionary change. Okay? So that was an assumption, but also one that was based on some evidence. Okay. So there were also a number of other kinds of criticisms that were actually sort of vicious towards this well, it's not a model. See, that's the problem. This is not a model, it's an observation, right? It's not a hypothesis, it's an observation. Now, let's talk. Okay, this was one of the things that they argued, people would argue, had to be occurring if you were going to get this sudden jump. So what could explain this pattern? Let's talk about how it could be explained actually on the basis of this model first, because that's what they did, is they took this peripatric speciation model of Ernst Mayer's and applied it to that pattern. Yeah?
It could be that the novel genotypes which arise, in the stasis, that is what is happening, when the novel genotypes arise, and they can occupy niches which weren't available to the ones before them.
Mm-hmm.
So that is when the radiation would happen,
Mm-hmm.
and then again after that, again it would get stabilized for a while till.
Mm-hmm. Okay. Yes. So their argument, first of all, remember, where are these data coming from again? Fossil record. Okay. How do we find fossils? Lots of different ways. Water cuts through an area, rain, they get exposed. But quite often, especially with microfossils, we find them because we're looking for oil. We drill, I know that sounds maybe silly, but it's not. If you are a petrochemical engineer geologist, what you've studied is paleobiology. Okay? You have studied where certain kinds of fossils occur in what time period. And the reason you've studied that over and over and over again and learned the fossil record backwards and forwards, particularly small fossils, is because when you go out with your company, like Pemex from Mexico, and drill holes all over the Yucatan Basin, you drill those holes and you lift that core out and you do sections, you're looking for what are called indicator species, okay? In other words, you're looking for fossils of certain kinds to tell you that it's really likely you're really close to oil, to big pools of what we want to burn, okay? So, because of that, because of the way we normally find fossils, that it's somewhat systematic as a petrochemical engineer geologist, but it's also idiosyncratic as to where we're going to drill those holes, you're much more likely to hit that than that, right? It's just probability. You're really unlikely, and this is what Steve Gould and Niles Eldredge argued. They said you're just not going to, when they applied this model, you're not going to find these in the fossil record, even if they are there developing these different genotypes, okay? You're just not going to hit them very often, which was problematic for this to some degree because their original argument was stasis, really seriously stasis, not some that were up here that you just didn't find. Okay? But what they also argued was in this model is that genotypically they may be varying all over the place, but morphologically they may not be, right? They may just not see it in the characters they're looking at. And so you get this hybridization event. They did not call it hybridization either, but you get this gene flow event here between two restricted forms. But what has to happen now also for it to go from here? What happens to this? Because remember, you're walking through time. You've just drug your core up from the seabed. And you're looking from early to more recent. And you're finding that here and that here. What happens to this then? Yeah, it has to go extinct, right? I mean, without Ernst Mayr's model at all, this has to go extinct. Or at least it contracts its range so much that you don't find it. Okay, so functionally it goes extinct. It never shows up again, so the logical conclusion is it goes extinct. So remember where I said that it could lead, you guys mentioned it could involve invading new niches, but also it can involve the displacement, driving this to extinction, the parental, the progenitor. It could be more fit in this environment than the progenitor. And lending credence to this are data I'll show you later in PowerPoints when we talk about hybridization. The fact that hybrids can be definitely more fit across many environments relative to their parents. Okay? Admixed genotypes have elevated fitness quite often relative to the parental types, or at a certain frequency relative to the parental types. Okay.
So that was the mechanism for computed activity.
This is the model that when Stephen Gould and Niles Eldredge got blasted for the hopeful monster, this macro mutation, which is not exactly what they were talking about, but it was to some degree. When they got blasted for that and said, you're crazy, that's not how evolution works. We all know it's microevolution. Well, we all knew that it was no epigenetics either, okay? So we'll see about that. But anyway, they argued, okay, macromutations are not an explanation here. And so Steve Gould and Niles Eldredge took Ernst Mayr's model later on and really applied it here and said, well, what we think may be happening could easily be happening is micromutation in here, still stasis, micromutation though, and this gene flow event that leads you to a new adaptive peak and also a new morphology. That was a way of them addressing the micromutations or microevolution versus macromutations. And also to explain why you would never find those that were going through, those organisms that were going through that kind of genetic drift and new genotypes that were arising during this time period. Well, because it's just unlikely to run across them. But see, what that leads to, I think, is that leads to the idea that maybe it really isn't stasis so much. Does that make sense? In other words, if you're going to propose that these new genotypes are arising and phenotypes are arising over time, but they're restricted in their geographic range, then you could end up having gradualism to some degree.
That's what I was thinking. This must express gradualism, but as you mentioned, the other phenotypes,
Yeah.
they are branching out, but they are quickly going extinct.
Mm-hmm. Yeah.
The overall effect is gradualism, really.
Yeah. So, okay. So the next question I have then is, how could we explain this through experimental error? Okay, these are not wrong data points, but what could be missing here? And this is what you're talking about. That could actually change that into a gradualistic, quote unquote.
Two fossils could be missing in between.
Yeah.
Because of that, we may think this is macroevolution, but in between there could be micro, and we may miss it, such that we can find it as macro.
But are you talking about that they're just actually not seeing the kind of evolutionary change because it's not fossilized? Or that we're just, see, these are two, what you're saying is exactly right. Okay, those are two explanations for the, or that's an explanation for these data. But there are two variables on, or two ways to think about this. So we may be missing actual data points in here, and this is something that people argued. You're just missing the data points in here to smooth this out. Okay, there are data points in here, you're just not seeing them because you're not hitting the fossils, you're not finding the fossils. That's the first argument against this pattern. This is an observation, it's not a hypothesis, but they're just saying you're missing all these dots in here that would smooth this out because they're more restricted, but they're very important in the evolution of these groups. The second part is, and people have asked this over and over again through these short courses for me, the characters they're measuring here obviously have to fossilize. Maybe there is a gradual evolution going on here in everything but these characteristics. The shell characteristics, the skeletal characteristics, whatever happens to be the hard fossil part. So people will say, what is going on with the soft parts? What's going on with the physiology? What's going on with the morphology? But morphology that we don't track here. Okay? If that makes sense. Okay. So either one of those are the case. Here's an, okay, observation, not hypothesis, but this is a very long time period. That's stasis. This is another argument against them having a real pattern here. What is going on with the environment during that extended period of time? Yeah, it's fluctuating wildly, right? I mean, we know, depending on where you are in the world, it really doesn't matter, but even in the tropics, it's moving, the environment is and climatic conditions are fluctuating wildly. So the question is, why do you have stasis if you have environmental dependent selection at all? So what is that? You know, why would you have this when the environment here is like the environmental fluctuations in here somewhere? Why wouldn't it have stepped up sooner? Does that make sense? So that's another. Once again, Jeremy, famous paleobiologist, works on fossil record, said, guys, that's the reality. That's what we see. And we have to explain why we see that over and over and over and over again in the fossil record. So, and I agree with him. If we're missing all those points, why do we see this so much? We miss all those points every time? You know what I mean? I mean, is it really true that most of the evolution is going on in small populations that we're never going to pick up on, and that's why we have these steps, and the big populations, no evolution? Maybe it is. And then on the other hand, though, why do you have stasis? We can understand jumps, big environmental perturbation, but why stasis across environmental perturbations? And that's where this comes back, where they would draw it back to this, where Steve Gould and Niles Eldredge would say, well, they are tracking the environment. We just don't see these small populations. They are tracking the environment. And this is just basically holding on. I don't know. It just doesn't. I need us to understand, though, this is an evolutionary biology speciation course, that this is the fossil, according to paleobiologists, this is the pattern they see. And we have to, as evolutionary biologists, we need to remember, as evolutionary biologists, evolutionary ecologists, whatever, we have two major data sources, right? Really. We have, and for a retrospective science, we have the fossil record and we have extant stuff to work on. Okay, now we can look back retrospectively now with genomics and that sort of thing to try to look at networks and understand migration, demography, and that sort of thing. That's a huge boon, but it's still basically a fossil record kind of survey in one sense. So even though there's a lot of questions about how this could happen, it is what we see in the fossil record, largely. Questions or comments about this? We've talked quite a bit about this. I think this is incredibly important for us to think about. But questions or more comments about this?
So, what do you comment that last sentence? I suppose this debate is not settled yet.
That it's not settled? Yeah, so going back to...
Yeah, we know that this occurs, but how?
I know. And so what is settled, and that's a great question. So is the debate really settled? Remember Doug Fatuma, 1989, I was teaching out of his textbook, and he said, look, there's still a debate on. 1 year later, going and hearing a keynote, Jeremy saying, there is no debate. If you're a paleobiologist, we know that's what we see, period. And then we've talked today, though, I want to get you guys to realize that we have to think about this rather than just accepting it either way. So where is the debate? The debate rests on, this is observation, why? What has caused this pattern? And so when we have paleobiologists come into our department occasionally and we get them to speak about their work, they have various explanations. First of all, they say, yeah, this is what we see. And then they have various explanations, models to test why they see this. Okay? Why they see this. Some of it dealing with maybe missing data, most of it dealing with biological attributes of what they're looking at. To the extreme of one of our paleobiologist colleagues who we had in, very well known, who studies mass extinctions. And he focuses particular attention right now on the KT extinction, when the 65 million years ago. And his comment to us in a journal club, he was really serious. He said, look, you guys study microevolution. He said, that's nice. He was a little patronizing. He said, that's great. We're like, oh, thank you. He said, another 10 million years, an asteroid's going to hit us. Everything resets, so your microevolutionary stuff doesn't count at all. Whoa, crap. I don't agree with him, but because I think the starting material actually after a mass extinction actually does matter. But his point was well taken. This could be just a meteor, right? That kind of an explanation of a stochastic-like event, extraterrestrial kind of event. It's not here, by the way. I'm saying that these kinds of changes in the fossil record, some of them may be that kind of idiosyncratic, the sea changes pH. And the organisms that they're looking at that are in the sediments now go extinct, and a new form is saline tolerant or pH tolerant, whatever it happens to be, at a certain level, and then it explodes. Okay, and so those are the kinds of models that they're proposing. The debate, microevolutionists like me go, I don't understand how this works still. And macroevolutionists or fossil paleobiologists go, well, we really don't care if you know how it works. It does, see? Like, yeah, but I like understand process. So that's where it stands. That's reality of what they see, but we're still trying to, I think as evolutionary biologists, deal with what might have generated it.
But then again, there are patterns of explosive speciation following every mass extinction event.
There are patterns of,
Explosive speciation, diversification
yeah,
following every.
yeah, absolutely. I mean, adaptive radiations, explosive radiations, whatever we want to call it, happen, 5 times. Well, hopefully they'll happen 6 after the ones that we're doing right now, mass extinction. But I worry for that one. But yeah, I mean, mammals after the KT. I mean, so yes, there are those. It's just, are those aren't necessarily punctuated equilibrium, right? That's not what they would be arguing. This is a lineage, a clade of organisms. And so yes, it could be looked at as punctuated equilibrium because you have dinosaurs gone, big jump, and mammals, replacing them in their niches. But that's not really what they're talking about here. This is if you go from here to up there, is it phyletic within a lineage, or is it cladistic, or more of a phylogenetic kind of jump, where you have a progenitor and then you change to a new? So a speciation event here. That's the other part of this model. This is where speciation occurs, according to their model, right? Whereas in Darwin's model, remember, you couldn't figure it out. Because where are you going to divide that line phyletically along a single lineage to determine where you have a new species? Where have you passed that point?
So Darwin did not really think in terms of branching here.
Well, he did. I mean, his one, yeah, he did. But I'll show you his one figure from Origin of Species is a phylogenetic, or is a tree, right? That's the only thing he put in there. So, but what he had were also, if this is present time, what he had were extinction events where they didn't reach there. But he had these kinds of tree. That's the tree that, kind of tree schematic that he did. And actually, it looks like it was hand-drawn, which it probably was, actually. But, yeah, so he believed in, or held to this kind of a model. But he was imagining that along these lineages that you had gradual phyletic kinds of change, right? And along here, you might have gradual phyletic change, have species, one species there, or well-marked varieties down here, and when it crosses this line, it's a new species, but you can't really diagnose it. So he had all of that overlaying on those both phyletic and what we would call cladistic evolutionary change. He pictured both, or described both. Okay, other questions or comments? How are we doing? About this, Myers, whatever else, before we move on. Yes?
Is it possible that the equilibrium represents the environmental stability, and after that punctuation, during the period of punctuation, there was sort of environment?
So are you asking if the environment is stable here? Yeah, it's too long of a time period. And that's what people pointed out. I mean, we're talking, in terms of, I don't remember how many tens or millions of years or whatever it is in here, but we're talking about time periods that greatly exceed, and should have episodes of major changes in environment and environment for these kinds of organisms that they're looking at. That's sort of important too, because some of the environments might have been stable for that long, probably not, but for these, yeah, they're fluctuating. If they're marine forms, they know that they're fluctuating in saline content, pH, episodically, periodically. They know the periodicity of that. So yeah, that's a major question. Why are they staying in stasis? And once again, people have argued, well, it's just because you're looking at hard parts. And if you were looking at the important parts, whatever those happen to be, then you would actually not see this kind of stasis. The counter to that is they don't just choose the parts they find, they know in many of these cases they have extant species, and they know what, like the swirl or twist on a shell and the openings, why they're adaptively important, why they affect fitness. And so their argument back is, look, the characters, first of all, we're looking at multiple, but the characters we're looking at, we know affect these organisms today, affect the fitness and the adaptiveness of those organisms. So we're not looking at neutral characters, is what their argument is. But anyway, yeah, the environment should, almost certainly, I don't even know how to put it, it varied. They look at paleobiological information about the environment. For stasis? Yeah, so their measurement of stasis, that's why they have these bars here. So these bars are here to make sure that these are all overlapping means, right, and standard deviations. But what those bars also do, demonstrate that these do not overlap with these. So it's actually a statistical methodology of just saying however many standard deviations you want to do, 95% confidence, 99, whatever I don't remember what those are, but that's what they're using. They're just saying, it's like looking for transgressive segregation in hybrids. Is the phenotypic space occupied by this putative homoploid hybrid outside of the phenotypic space by however many standard deviations of the parental forms? That's what they're saying here. Not that these are hybrids. Not that they're arguing that those are hybrids, but that's the kind of measurement. Yeah?
How likely do you think it is that, is it believable to think that every time that a hybridization happened, it was from the parental population and drives this extinction?
No, so question is what is the frequency, what is the likelihood that the parental form goes extinct due to the hybrid forming and displacing it or outcompeting it? I mean, if we look at neo-hybrid species, like homoploids or neo-allopolyploids, you don't see parental progenitor extinction. Okay, you see the formation of a new homoploid hybrid species, Heliconius butterfly, and it's a part of that formation.
Counted in here though as the same species, right? If someone is making a graph like that.
What Stephen Gould and Niles Eldridge argue here is that's a species and that's a species. This is a speciation event. Speciation is not occurring here. This is a part of their model. Speciation is not occurring here. It's occurring here. Oh, sorry, here. And the loss of this means the extinction of that. Now, I think we can agree that if this disappears from the fossil records and never seen again, it's gone extinct. The question is why. But with that model, when they applied that model, what they were hypothesizing was you're losing this because of this. There's a cause and effect.
And that was only one of the explanations. That was only one explanation to explain this object.
When they applied this model, yeah, exactly.
In that model, when, for example, alveoloid form, like they are more likely to not drive the parent population.
Yeah, what we see in, like I say, in neo-allopolyploids, neo-homoploid species, because that's a speciation step. And so if that model is correct and there's a hybridization, let's say here. I know Meyer didn't call it hybridization, but you have a hybridization here. If this hybridization event is like neo-hybridization, hybrid speciation events, we wouldn't predict necessarily the loss of that or the loss of both parents in the case of, you're losing all of those. So no, I'm telling you what we observe in recent ones. In actual fact, what we observe throughout clades, from ancient times, in introgressive hybridization, we don't see the displacement and loss per se of parental forms. We see material moving but not losing biodiversity. Increases in biodiversity, yes, but not losses. Sorry, so you were saying that they didn't just have this as an explanation. That is absolutely correct. Right. And if you read that, you'll realize that they didn't really talk about that here. This is but I wanted to tie that with this because later on they melded these two and argued that that could explain the microevolution that they weren't showing here or weren't detecting here. Somebody else had their hand up. I'm sorry. Okay.
So when you are with this data you are comparing with this model.
Mm-hmm.
So I think we are assuming this novel genetic norm that is as like this that is that hopeful monster. So if that is so now as you told when there is the equilibrium, that is a long period of time.
Mm-hmm.
So within that equilibrium also the environment is not same, it is undergoing massive changes.
Mm.
So if it is undergoing massive changes there also, so there should also this kind of event happen.
Yeah.
So again, like in between also we should get this novel genetic norm.
Yes.
So if we get in between also this norm, then in between also this jump should happen.
Right.
It is not happening. Is there an explanation for that?
Yeah, you're talking about in here we should be seeing these jumps. Yeah. The explanation is they don't know why. Once again, across these, if that model is correct, and you're right. By the way, the reason they called it a hopeful monster, people use that terminology pejoratively, negatively, was that if you had this jump and you're up here by yourself looking for somebody to mate with, you're in trouble. You're a hopeful monster. You're hoping somebody else reaches that at the same time, right, of the opposite sex. That's what they argued against Goldschmidt's. Hopeful monster. You hope somebody else gets the same macromutation at the same time. Now, why it doesn't bounce around here, if this model is correct, once again, what they argued was, well, it was bouncing around, but it was in limited populations. This is what Gould and Eldridge later applied and said, these peripheral populations that's where all the evolution, microevolution is going on. Then there's this gene flow event and you have that jump, but you don't see any of this microevolution going on because it's in small populations and we're not picking up the fossils. We're just not finding them. But why I think that weakens this macromutation or whatever we want to call it, speciation step is, well then that means the speciation steps are going on in here, we're just not seeing it. So doesn't that smooth that out, potentially, and turn it more into gradualism than this? Okay.
Sorry, I was just totally lost. Were we supposed to have read this paper yesterday coming to the class?
For today?
Yeah.
Yeah.
Oh, I missed that completely. We didn't get the papers either. So,
What? Oh, okay. Sorry, this is fossil evidence. Did anybody get this paper? No? Oh my. So did I not give it to you guys? Okay, wait a second. Hang on.
And we have the Dropbox that you sent me yesterday with the
Say again.
Dropbox.
Yeah. Okay, so let's go through this because this is incredibly important that I know what you guys have and what you don't. Okay, so did you get, let's just go through, did you get the papers that we talked about yesterday? You haven't gotten anything. Oh my. Well, I apologize greatly. I thought we set up a Dropbox. Okay. We will set up a Dropbox so that you have these papers. I really apologize, guys. You did a great job of discussing papers you didn't read yesterday, and you have today as well. Let's take, it's 11. Let's take a break. We'll come back. I'll talk to you a little bit about this, and then we'll figure out what exactly I did wrong, because I thought we had you set up for a Dropbox with the PDFs. So we'll get them to you. Yeah, we'll get them to you. We'll have the information on the, hang on just a second, let's get here. So, yeah, tomorrow I'll be lecturing, so you'll be safe there. The question is, and I was talking to Hema yesterday, we're using my book, and there are a few copies of this. We're using my book. Once I get back from Pune, I'll give one more lecture here, okay? But then we switch over to this, and the question is how they're going to get that, you know?
We have limited copies in the library.
Mm-hmm.
I think 2 or 3 copies.
Okay.
So, is it possible that some chapters can be shared with them as PDF?
Not really. I was specifically told that was a violation of copyright by my publisher because I asked him if I could do that. The question is whether or not, and this might not be appropriate either, but I'll just throw it out there as a possibility. Is there any possibility of photocopying this thing, which is what they did in China? I don't know if you guys can do that. I know it's a lot of material.
So, is it specific chapters or do you want everything?
Well, they're going to be reading 1 through 8. We're going to be discussing 1 through 8. And I do not have lectures enough to cover all this time. Although we've been doing real well with them not reading the material so far. Shoot. I am so sorry, guys. I guess I slipped up and didn't send this. I thought we had set up a Dropbox for y'all. But we will do it. I'll get the papers, even though now it's retrospective, obviously, but we'll get all of the readings in there so that you'll have them as resources for later on. But we'll also get the, I don't know what to tell you about the book. It's what you and I had discussed before. So,
Maybe we can talk about it.
Okay, break time. And we'll come back in just a little bit and we'll finish this.